yukhrijul hayya minal mayyiti wa mukhrijul mayyiti minal hayy. Dzalikumullahu fa anna tufakun. Faliqul isbahi wa ja'alal layla sakanan wasyamsa wal qamara husbana. Dzalika taqdirul azizil alim. Wa huwa alladhi ja'ala lakumun nujuma litahtadu biha fi dhulumatil barri wal bahr. Qad fassalna al-ayati liqaumin ya'lamun. Wa huwa alladhi ansha'akum min nafsin wahidatin famustaqarrun wa mustawda'. Qad fassalna al-ayati liqaumin yafqahun. Shadaqallahul azim. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Ms. Desti Siantika, for reciting the Quran. For the next agenda is singing the national Indonesia anthem, Indonesia Raya. Kindly, all of participants, please to stand up for singing Indonesia Raya. participants, please be seated. And next, I would like to invite the Muhammadiyah University of Pringsewu student dancers. The dancers will dance Sigah Pengunten. Sigah Pengunten is traditional dance from Lampung which is performed to welcome important guests, whether local guests, government officials, and the other important people. This dance will be performed by Miftahul Jannah, Naina, Zahra Putri Anjani, Tinoko Abela, Rofi. Please welcome. officers, ladies and gentlemen, now I would like to invite the Vice Rector for Academic for Muhammadiyah University of Pringsewu to deliver her welcome remark and to open this International Symposium 2023. To Dr. Triyuni Hendrawati, MPD, the stage is yours.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Let us praise Allah Subhanahu wa Ta'ala for giving us endless blessing, so that we all can be here together in this special occasion in a perfect condition and health. May salawat and salam are always given to our Prophet Muhammad Sallallahu Alaihi Wasallam, who has taught their people respectively in order to attain happy and prosperous life in this world and hereafter. It is such an honor for me, Triyuni Hendrawati. Welcome to second symposium on advanced preclinical research and clinical trials. On behalf of the Rector University of Muhammadiyah Pringsewu Lampung, I would like to deliver his apologies of his absence in this event and his big thank you for all. I'm very thankful to you, Associate Professor Dr. Vetri Salven from Monash University, Malaysia, and lecturer of Mahsa University, Malaysia, Professor Bikas Mehdi from the Postgraduate Institute of Medical Education and Research, Chandigarh, India, and all of great speakers for presenting here with us today despite your busy schedule. Very thankful to you, co-hosts from Unila, ITERA, Stikes Adila, Poltekkes and University of Tulang Bawang, who have joined together to organize this activity. Thank you to my college, Vice of Rector University of Muhammadiyah Pringsewu, Mrs. Asri Rahmawati, and Mr. Fatoni, and the Dean of Faculty of Health, Mrs. Elmi Nuryati, who can be present in this occasion. I also wish to thank all to the committees who have organized this wonderful annual event. I am especially excited to welcome the participants who take part in this international symposium. Without you, it is impossible for us to be in this beautiful place together now. Mr. Vetri Salvan, Mr. Bikas, and all invited guests, for you to know that currently University of Muhammadiyah Pringsewu have 5 faculties: Faculties of Health, Faculty of Education, Faculty of Sharia, Faculty of Economic and Business, and Faculty of Computer and Science. The 5 existing faculties, we manage 16 study programs. Finally, I wish you a happy holding of the symposium. I hope it will be useful for increasing our knowledge and friendship. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you for a wonderful speech. Ladies and gentlemen, now we have come to our, ladies and gentlemen, before our start to the main agenda, I would like to invite our speakers and our co-hosts and the Vice Rector and Vice Rector for Academic, Vice Rector for Human Resource and Finance, and Vice Rector for Students Affairs and Al-Islam Kemuhammadiyahan to come in front of the stage to take picture together. And I would like to invite representative by Unila, Lampung University, Sumatera Institute of Technology, Tulang Bawang University, apa lagi? Politeknik, Tanjung Karang Politeknik, Stikes Adila, siapa? All of the organization profession, the Dean of the Faculty of Health Muhammadiyah University of Pringsewu, the Dean of Education University of Muhammadiyah Pringsewu, the Dean of Economy Faculty, the Dean of Sharia Faculty, wakil dekannya, wakil dekannya. Sudah. Thank you for the time and taking picture with us. Now please be seated on the chair that has been prepared. Ladies and gentlemen, now we have come to our main agenda. A presentation and discussion will be moderated by our moderator, Mr. Bayu Anggileo Pramesona, PhD. Okay. Mr. Bayu Anggileo Pramesona, PhD, is the lecturer of Lampung University. Ladies and gentlemen, please welcome Mr. Bayu Anggileo Pramesona, PhD.
Oke. Assalamualaikum warahmatullahi wabarakatuh. Tabi pun. Okay, I'm so glad to be here as your moderator today. Yeah. Good morning, ladies and gentlemen, once again. Yeah, first of all, I would like to welcome you all to our second symposium on advanced preclinical research and clinical trials, or APRECT 2023. Yeah? And for this year, the highlighted topic is complementary therapy as new treatment in health sector, ladies and gentlemen. And as we know, that complementary therapy is becoming increasingly as popular. More people seek out a natural or non-invasive approach to healthcare. Yeah, some studies found that in Indonesia, Indonesia during COVID-19 pandemic was the most country that used herbal medicine as a complementary treatment, and also another country like Saudi Arabia, Vietnam, and also the Hong Kong as well, using the herbal medicine as treatment for like complementary therapy in COVID-19, during COVID-19. But I just wonder, the big question is how, how does pharmacology see complementary treatment or therapy as an alternative or a new treatment in health sector? And how safe is it for treatment? And to answer those questions, we have invited and already joined with us 3 honorable speakers for first panel session. They are Professor Bikas Medi from Department of Pharmacology, Postgraduate Institute of Medical Education and Research, India. And second one, Ms. Apotheker Dwi Aulia Ramdini, M.Pharm, from Department of Pharmacy, Universitas Lampung. And the third one, Ms. Apotheker Nisa Yulianti Suprahman, Master of Science, from Department of Pharmacy, Sumatra Institute of Technology, or ITERA. So I think I would like to invite all those speakers to move to the stage, please. Prof. Bikas, Ms. Apotheker Dwi, please welcome to the stage. Big applause, please. Okay, thank you so much. And also Ms. Nisa Yulianti Suprahman. Mrs., I guess, yeah? Okay. Before we start, please let me introduce our keynote speaker for this morning, Prof. Bikas Medi. He is professor at Department of Pharmacology, Postgraduate Institute of Medical Education and Research from Chandigarh, India. He served as editor-in-chief, Indian Journal of Pharmacology, and also International Journal of Pharmaceutical Science and Nanotechnology. He also former additional medical superintendent, founder of Experimental Pharmacology and Neurobehavioral Laboratory, and coordinator Postgraduate Institute Medical Education and Research, Pharmacovigilance and Material-Vigilance Center, Regional Coordinator, North Zone Ministry of Youth and Sport, Government of India, Co-Convener India Initiate Program, former Secretary Clinical Pharmacology of Indian Pharmacology Society, and he got many awards such as Dr. D.N. Prasad Memorial Award, Dr. V.K. Bagrava Award, Dr. B.N. Ghosh and Kulramnath Chopra Oration from Indian Pharmacological Society, FIDA, in 2023. He also dedicated in 2% scientists across the world conducted by Elsevier and Stanford University, nominated as member executive committee of IUPAR for global coverage IUPAR activities in coordination with WHO from 2023 until 2026, and member of Codex Committee. He has published 5 books and more than 5 research papers, yeah, Professor. And so without further ado, I would like to welcome Professor Bikas to deliver his presentation. Please, Professor Bikas. Okay, wait a minute. Namaste, Prof. Bikas. Namaste. Welcome to Pringsewu, Lampung.
Okay, good morning, everyone. Good morning. Yeah. And I like to thank you, the organizer, for giving me the opportunity to speak to you. So as you can see that we planned this program as advanced course in preclinical and clinical setup. But here it is very important point is that how we can promote the complementary medicine. We know that we are familiar with, modern medicine, but how modern medicine has been practiced, or you can incorporate this knowledge into the clinical practice. So, moment you say complementary and alternative medicine, so that means you know that we have a rich, you know, source from our civilization. So you can see when you go to the next slide, how the modern medicines are practiced since 5,000 years back. So you can take an example of yoga, acupuncture, then you have a diet therapy, Reiki. There are several practices there into the alternative medicine. Now, moment you say alternative medicine, you can say, what are the domain of alternative medicine? So you can say that, you wanted to manipulate body and mind. Second point is, if you say mind and body manipulation. Okay. So you just think of how different complementary medicine or alternative medicine could be. If you say manipulation of body and method, can you do it? Okay. Second thing is you can do mind and body manipulation or alternative medicine system. So there are several systems out there, like where you introduce or manipulate the energy. And third, if you say that can you develop a medicine or a product which is biologically approved therapy. So here, if you look at that, time for therapy. So what is the time for therapy? If you can say that nutritional, okay, then can we have an impact on when you plan for nutritional therapy or psychological or physical, or you can say, can you combine all the therapy together so that we can get a maximum, efficacy or in terms of safety also. Now here if you see that we talk about complementary medicine or we talk about the alternative medicine. Now when you say about complementary medicine practice that it conjunction with Western medicine. Now second thing is when we develop this complementary medicine, we always say that can we develop in terms of evidence-based, whatever it is required. Any treatment you recommend, it has to be with the data that can you generate this data. So that is why to avoid any traditional one, if you say this is scientific approach has been made. So another one is alternative practice. You have a lot of practice we have been practicing over a period of time, and that is why you can say what are the domains of complementary and alternative medicine. You say alternative medical system, that means we practice like in India we have Ayurveda, Chinese traditional medicine, like that we have in Native American. If you look at Egyptian, civilization. So there are several practices all over the world. Second thing is you talk about mind-body intervention. So here if you see that you talk about any approach is there, can you manipulate that cognitive and behavioral one? Third thing is biologically based therapies, we have a lot of product is available. And fourth thing is manipulated body and method. So there are different methods. Now coming to some of the oldest one, when we talk about the oldest 5,000 years back, we practice Ayurvedic medicine. There are a lot of products is there where you practice. So if you look back in history, 5,000 years back, we used to practice Indian medical system, which is indigenously developed. Then even 300 years, there is a acharya called Charak, and he prepared all the pharmacopoeia, whatever the treatment is there. So that is why if you look at the today's modern medicine, you say you have a ENT, there are 56 departments is there. So similarly, during that time, Ayurveda, it was divided into different parts, medicine, surgical, even ENT, ear, nose, and anything which is surgical or 8 part, which management or treatment of any sexual dysfunction. So fundamental principle, if you say that, some of the basic of Ayurveda, so you can say that India has created a department or ministry called Ayush. A, it is for Ayurveda. Y, it is for yoga. Third, U is for Unani medicine. And S is for Siddha, or H for homeopathy. So this is how, we have Ministry of Ayush who look after all the complementary medicine in order to promote into the practice. Now, when you look at any of the medicinal plant, you know that earlier setback was that it was not developed as per the modern medicine. So whenever you say that this particular product has an effect, so people used to question that what is the evidence you have in order to establish its efficacy and safety in a particular disease. So that's how that World Health Organization is said that any plant whose one or any of the more organs substitute used for therapeutic purpose or synthesis for the drug. So basically if you say that a plant product it work. So here if you say that is it because of the leaf, is it because of the bark, or is it because of the root? Okay, so then you go in more detail that which particular ingredient it is work. So that's how, 80% of the people in developing country they use, like we have a lot of example of anti-cancer therapy, which is isolated from the plant. You can take an example of anti-malarial drug, like it is from the Artemisia. So we have many examples. So that is why if you look at, how many plants it has been developed and it has a diversity. So you can take an example that Indian system, Ayurveda, Unani, homeopathy, yoga, siddha or AMSI, there is a new branch has come. So altogether if you see that how many product has been developed, so maximum share is 28% is Ayurveda. So similarly, when you go for Ayurvedic system, it is actually at a very old, okay? And if you can see that, what is the basis? Basis is it says that, yar, bile and phlegm. So these are the principles, three principles it is developed. So when you look at this, that before you give the treatment, you wanted to make a diagnosis. And based on the diagnosis, so normally in modern medicine, what do we do? You make a provisional diagnosis. Now we have a lot of investigation now. So after that, when you go for a biochemical test or radiological test, then you make a accurate diagnosis. Once the diagnosis is made, then you have a standard treatment plan. So accordingly, you use the drug. So similarly, in earlier time also you make a diagnosis, and based on the Vata, Pitta, Kapha, air and other principle, you are going to make the treatment or give the treatment. And that's how the Unani also has been started. So if you look back and see that how Unani has been started from the Arabs, there is a Unani province where they used to practice, and that's how they say that what about the principle if you say element, humor, the body fluid or the vital spirit, then you have all the treatment, and temperament on organ and the faculties for power. So this is how you do Unani treatment, and they also make the diagnosis and then followed by the treatment. So here, if you look at the pharmacotherapy, this has been practiced for years together, okay, or decades together. And based on that, you give that exercise, rest, or, sleep, adequate sleep, and also psychological behavioral manipulation, and accordingly you give the, management. Now, what about homeopathy? Homeopathy concept actually start from the Germany. So basically, the one of the renowned person who said that because of the medicine, there are a lot of adverse effect is there. So he wanted to titrate with a lower dose, and then you give the treatment. And that is why you see the direction of the cure with a minimum dose, and the law is single remedies for a chronic disease. So any chronic disease, if you give a low amount, and it can cure. So here also if you see that they make a diagnosis, they make a treatment with a serial dilution or succession or titration. So these are how you manage the patient with making an assessment, then you go for the investigation, making a diagnosis and give the treatment, appropriate treatment. So here, one of the very successful, complementary therapy or alternative therapy is Tibetan system. So in Tibetan system, if you say it is similar to Ayurveda, it is a Buddhist tradition, and here it is more than 2005 years old. Okay, now when you look at the Ayurveda, it is more than 5,000 years old. So here if you see that they make the treatment, even some of the rare disease, I've seen people suffering from sarcoidosis, which are being managed with, Tibetan system. So this is what the principle is: earth, water, fire, wind, and space. And based on that, they make the assessment for the treatment. So this is what the element of Tibetan medicine, what I say: earth, water, fire, wind, and the space. So this is how they make a visual diagnosis. Those who are expert, then they make a touch diagnosis or questioning diagnosis. So idea of giving the treatment, can you go for a diet modification or health medicine or physical therapies? So these are combination of the therapy it is used. So here if you see the formulation they make based on all the principle, earth, fire, or other principle. So these are also, you can look your tradition, Indonesian traditional system, where you have a combination therapy, massage therapy, and other therapy. So these are also very, very old. So if you look at the basis of, it has been written in the Ramayana book, chapter 1:9, how different therapy can be introduced. So these has been practiced, and you have a product which has been taken from plant, animal, minerals, or different abstract is there. So these are, several, ingredients are available in Indonesian traditional system. And along with that, you have a combination therapy for traditional massage. So these has been practiced. So this is what, technique is there. Now question here is, how much we are successful making a traditional preparation? So if you look at that, so many product has been, isolated from the traditional system. And it has been used in various diseases, like it has been isolated from root, tuber, rhizome. So there are several products has been isolated and it has been marketed. So these are the product in Indian system. We have a large company who are working like Himalaya, Natural Remedies, or the Dabur. So these are the company. So if you look at some of the product, we take them from the plant, then the fruit, and we make the product, final product or finished product is face oil or nutritional capsule. So these are some of the example how different product is marketed and it has been used for prophylactic and preventive disease. So these are the product, if you look at, leaf 52. So this has been used in case of, liver toxicity or in order to improve the liver function test. Now, similarly, Chyawanprash or Cystone, where it is used for whenever there is a problem with the kidney stone to dissolve the kidney stone, and these are some of the product. Now, question here, as a medical student or healthcare workers, what is the justification for use and how do you extract this material? So these are, if you have a plant powder, so you can use a simple instrument for extracting water extract or methanol extract, and then you can make a concentrated form and you can use it. So these are the process, even if you use like any plant you collect, it is important that if it is a rainy season or if it is in a winter or the summer, this make lot of difference in terms of medicinal properties. So you can collect and you can make an essential oil, and finally, so these are the process. Here it is very important to know that if you prepare, why you are getting a pharmacological action. Okay, so that is why you have a process to identify the active ingredient by LCMS or SPTLC. So these are some of the example, how do you identify the ingredient, active ingredient, where you can link to that later you can think of semi-synthetically synthesis the molecule. So these are process for analytical technique. You can go through any of the plan when you identify based on the earlier data. So this how you can do it, and these are the technique is available, analytical technique. But ultimately you know that any product you develop, so what you do is first you look at the animal toxicity study. Is it safe in animal? Okay, or is it safe in smaller animal and higher animal? So later you can do that, you go for a first time in human, the phase 1 study, then you go first time in healthy volunteers, phase 2 study, then you go for larger, human, and it is in, phase 3 study, larger clinical study. But you might have seen during the COVID, how many drugs we repurpose. So if any emergency take place, we also go for a accelerated drug development process. And that is why many drugs like hydroxychloroquine, ivermectin, it was repurposed. So that's how we have a process for synthesis and purification. Then you go for a short-term studies. Like if you use the drug for 7 to 10 days, you need to go for an acute toxicity study. But if you go for a 30 to 40 days, you have to go for a repeated dose toxicity study. But if you use for the drug like diabetes, hypertension, we use the drug for the years together. So then you need a long-term toxicity studies. That is almost give the drug and you evaluate the animal, rat and mice, for almost 2 years. And that's how you divide. So if you look at that, what are the animal you are going to use? Now, if you remember thalidomide, when it was introduced into the pregnant woman, and there is a lot of cases of phacomelia. And that is why now we have a concept, any drug you use, is there any species variation? Okay, so that is why initially when you are going to test a drug, you are going to do it in a smaller animal. And then later in a higher animal, smaller animal in mice and rat, and later in a higher animal in monkeys and in dog. So that's how we have different models. So when you work, you have to see that can you generate the proof of concept study. So how do you do that? You do an in silico study. After in silico study, you do an in vitro study. Then you do a particular model where you do an in vivo study. You generate the in vivo proof of concept study. After that, what you are going to do is you are going to do a regulatory toxicity study. So these are the models are there. Now, as you can see that most of the time when you say we use the mice 70% or the rat, almost 30%. So because it has a similarity. So when you plan for any preclinical study, initially you go for a rodent, then you go for a higher animal because you wanted to find any variation in terms of species. Okay, so that is why now we talk about any individual variation also from human also, that one person they develop very good efficacy, another person they develop side effects. So that's how you have now lot of people they encourage that we should not use animal. So now US FDA also says that can you use cell-based method to evaluate the toxicity of the drug. So we have a lot of alternative therapy or cell-based cell line where you can evaluate the drug toxicity and efficacy. So these are methods. Now look at this. During the COVID, we have categorized how many alternative therapy can be effective in COVID-19. So these are, we could find that these are effective in COVID-19, and you can see that how, approaching the ethnopharmacology approach, you can do the clinical trial with traditional medicine and establish. And that is why during this COVID time, number of experiment was, it was increased. So if you look at the earlier one, number, whatever it is in the rodent or the higher animal, it was tremendously increased. So altogether, if you see that any use of the drug in animal, what do you want, that what about the drug, any cosmetic, medical device, nutrition, everywhere we use the animal experimentation. But more important point is when you do the study, you have to do under good laboratory practice. And this good laboratory practice is a quality system where you have to generate the animal toxicity data, and this is actually tell you that how do you plan the study, how you are going to perform the study, how you are going to monitor the study, and how you are going to record the study, and how you are going to archive the study. Normally, animal toxicity study, you are going to keep it for 9 years. We call it 3 cycle. So altogether, if you see that whether it is a herbal product, pharmaceutical product, pesticide, or any nutritional food, also you have to do the GLP. So a lot of people, like they say GLP is only applied in the lab. It is not true. If you go to tea garden or paddy field, so there also GLP is applied. So GLP basically you wanted to evaluate the best quality data, okay, or so that you can say that you can go for an audit or you can go for verification of the data. So that's how, if you do not have any GLP, when you plan the study, you can say that you have a hypothesis, you write the protocol with well-designed protocol, then you have a test system, or people who are doing the study are trained, and all the methodology, you can say that SOP, standard operating procedure, you follow. So ultimately, you generate data which is at par with the global level. So that's how we do. So this is what some of the basic point, that are your people who are doing the study are trained. Do you follow that SOP? Okay, you have an SOP for SOP, and you have a other SOP also. Then you can say that you have a quality data, and all the facilities are appropriate, and that is how you say all the rules are followed as per OECD, Organization of Economic Cooperation and Development, and all test system, like animal system you use, or any chemical it is used, it is characterized. So that's how it is allowed that you can do that all characterization, and all the pharmaceutical product, all the pesticide, all the herbal product or complementary medicine, you can say. So basically, any product you are going to test in animal testing facilities, you need to know that how the product is absorbed, how the product is distributed, how the product is metabolized, and how the product is excreted. So that's how the GLP principle is applied in terms of that how you are going to organize the plan for a study, and you write the protocol, and you incorporate that SOP, standard operating procedure, and you generate the data. So ultimately, you are going to submit the report to the regulatory authority who approve the product. So that is why quality assurance is very important, how you are making the quality data in terms of quality control, because in this system, you have a quality assurance program, and ultimately you are going to keep the data for almost, for if you are in drug development process, 12 to 15 years, and so that anybody can look at the data if when you plan for a clinical study. So there is a weakness because you need a lot of money, but ultimately strength is that you can say that I have done the study under GLP, good laboratory practice, you can come and verify it. And that's how once you do preclinical study, you can go for the next phase is clinical. So there is a guideline from Indian government how you plan for a clinical study in case of a complementary medicine or any product. So these are the guidelines are there. Ultimately, you know that whenever you plan for any clinical study, you have to do it under GCP conditions. So these are the guidelines are there. You can just go through. So how you look at the product, the preclinical data, how you plan for a clinical study. So these are, when you plan for a clinical study, you are going to follow the GCP, good clinical practice. So internationally, what you are going to follow is, ICH GCP. So ICH GCP, when you plan, person who does the study, okay, they have to follow this guideline, ICH guideline. So it will tell you that, what is your responsibility as an investigator? What is your responsibility who sponsors the study? And we have an ethical approval. There is an ethics committee who approved the study. So altogether, if you see that all, regulators sponsor, then ultimately patient who participate in the study, they have to fulfill that altogether regulator will make sure that this study is done ethically, or, regulator also sees. So ultimately the principle is that, as I say, when you talk about guideline, it talk about that it has a responsibility. If I do a study as an investigator, I have a responsibility. If I approve a study as an investigator, I have a responsibility. Okay, ethics committee has a responsibility. So all 15 key responsibility is followed in ICH guideline. So you say, you write the protocol. What about your, standard operating procedure? What about your system? Okay, support system. So ultimately, if you say that if you wanted to do a study, are you qualified enough to do the study? Like especially in herbal medicine or alternative therapy, modern doctors is not allowed unless you have an investigator from, alternative therapy. So all ethics committee, they review. So normally when ethics committee, they review the scientific part as well as ethical part. So ultimately, any problem is there, okay? So any adverse event is there. So you are going to report to ethics committee and to the regulator. So ultimately, you are going to submit the data to the regulatory authority. And that's how, based on this data, whether data as per the quality data or not, and here, while doing the study, you are also going to register in one of the registry. Every country have a registry. Like in India, we have registry. US, they have a registry. Now for alternative therapy, we start a portal, new portal. So ultimately, when you do a clinical study, you are going to look at, all preclinical data. You write the protocol, design of the study, then you look at the safety aspect, and ultimately, while completing the study, you are going to archive for almost 5 years. Preclinical data, you are going to keep it for 9 years. Clinical data, you are going to keep it for 5 years. Altogether, maybe 15 years. So there is a, regulation by every country, drug and food regulation. So here, if you say that any herbal product, now Indian regulations say that you have to identify 4 active ingredients before you wanted to make, do the study, either preclinical or the clinical. So when you, manufacture this product, now any product, when you wanted to manufacture for human situation, what you have to do it, you have to prepare in GMP condition, good manufacturing practice. So that is why you need a test license for this. So this is the process, but in case of a food, also regulation is there. So how the food is applied, especially the medicinal value for the food. So that is why we have a process for scientific panel, committee, food authority, they also give. So any doctors who approve any of the food product or medicinal product, that has to. So you can say that there are a lot of product that is available, okay? So this has been used, like garlic has been used for hypercholesteremia. But it is very important to look at that when you look at the advertisement for a food product, like this is for a food, weight reduction. We have to critically look at, how the advertisement is there. So this is what we have to see that, is it after doing the study, they have advertised that there is a scientific study has been done. So this is what. But ultimately, when you talk about, any of the natural product, it is better than any of the modern medicine. So regulator also have, public relation office. You can go and discuss with the data, get an approval. But ultimately, you know that in pharmacology, you have to do the research. There is an industry component. The students should be trained in how you do it in industry. Then if you see that we have, like we contribute in drug development, okay, we contribute in pharmacovigilance, means any product you market, if there is an adverse effect, you are going to record. So we have a program. So similarly, healthcare services, other. So we have a lot of key role to play in this. So what are the pharmacologists' role? Maybe medical education or in, national pharmacovigilance program, and then all the regulatory affairs, marketing, community, pharmacovigilance, toxicology. There are a lot of key roles. So any policy decision take by the government, if you can contribute, what should be the medicine available? Like we have a concept of essential medicine. So that means every country, they prepare the list of medicine. Like WHO has prepared in 1997, but before that, Tanzania has prepared the essential medicine list. So if you can incorporate the list in every provinces and every hospital, we have a list of hospital formulary, what are the medicine is required, then it will be very successful that you can contribute. Now, we now talk about a quality delivery. You might say that we have been looking around 100%, but what about quality? Is this delivery is person-centered? Is this delivery of medicine is safe and effective? And that is why it is very important that if you look at that your healthcare delivery, it's maybe you have a sophisticated instrument, but if you say somebody falls sick, if you say, are you fine? Do you need anything? And this is what the important is in the hospital. But at the same time, if you say, quality of medicine is very important. How do you check the quality? Every medicine is marketed. It is at par with different pharmacopoeia, like Indonesia pharmacopoeia, British pharmacopoeia, USB. So if it is not at par with the pharmacopoeia, you are going to call it not of standard quality, spurious or counterfeit. But at the same time, when you look at the prescription, when you look at the prescription, like we now have an e-prescription. So there are, WHO criteria for looking at the prescription. How many drug has been prescribed? Okay, is there any fixed dose combination? Is there any antimicrobial? Okay, is there any injectable? So we have a software now. We train the people with 47 modules in India for prescription skill development program. But altogether, if you say that when you work with different national program, okay, like malaria program, filaria program, with all the people together, definitely we'll have a successful, therapy. How you manage in primary health center, how you manage in secondary health center, how you manage in tertiary care center. But one of the important component is we have lot of scope to develop alternative therapy into modern medicine. But what I say that whatever you develop, it has to be evidence-based. So if you do a preclinical study, that has to be a GLP compliant study. And after doing that, when you plan for a clinical study, you say, I follow ICH, International Council of Harmonization for doing a clinical study. And that's how you'll be successful. So this is one of the recent book has been released. You can read that. And all this you can see that every country has a national health policy. And based on the national health policy, all this program has been implemented. But it is important to have a documentation. So if you ask, you read about Robin Hood, he did a lot of service. But if you ask, did you have a receipt for giving the service? He doesn't have a receipt. So today's era, you ask for all the documentation, whatever you do, preclinical or clinical study. But here it is very important that as a university, what is your contribution of your original study? How many preclinical study you have done? How many clinical study you've done? If you take an example of Switzerland, smaller country, they are contributing almost 39 to 40%. So that is why all the faculty members and a student must concentrate on research. Can you generate, some of the original study and contribute? So with this, I'd like to thank you. And as you can see, that we need to do a lot of work in terms of original study, but we need to look at the international guideline and national guideline to generate the data. So this is my lab. Thank you very much. Any questions you have? Thank you.
Okay. Thank you very much, Prof. Bikas, for the very insightful presentation. And maybe could we move to the next speaker, Prof?
Yeah, fine.
And then we will do our panel after all of the speakers already presented the presentation. So for the next speaker, I would like to invite Mrs. Apotheker Nisa Yulianti Suprahman, Master of Science. She was born in Bandar Lampung, July 6, 1991. She graduated her Bachelor of Pharmacy degree from Universitas Indonesia, such as an NMDA receptor inhibition in osteoarthritis, immunomodulatory effect of vitamin D and phytochemicals, in silico screening, synthesis and in vitro study of serum bone and its derivatives as anti-cancer candidate for breast and colon cancer, and in silico exploration in vitro and in vivo study of gelatin C as anti-cancer candidate for breast and prostate cancer. She also has published some papers, both in Indonesian, national and international journals. So, please big hand to our second speaker, Apotheker Nisa, Master of Science, please. Your time will be 30 minutes, I think.
Assalamualaikum warahmatullahi wabarakatuh. The Honorable President of Universitas Muhammadiyah Pringsewu and Vice President of Universitas Muhammadiyah Pringsewu, Dr. Tri Yuni Hendrawati. The Honorable, the other speaker, Dr. Fetri Selvan Subramanian and Professor Bikash Medi from Postgraduate Institute of Medical and Education Research India and Mahsa and Monash University. the Honorable, my fellow lecturer from Lampung University, Institute Technology Sumatera, Health Polytechnic of the Ministry of Health, Tulang Bawang University and Adila School of Health, my beloved students that present here today in this beautiful city. I'm very pleased to speak here in front of you to share a bit of drug discovery perspective under preclinical study, especially on the topic of osteoarthritis. Okay, today I would like to share a bit of what happened globally on drug discovery of osteoarthritis. Check, check. Oh. Check. Can you hear my voice? Okay, thank you. So, the Honorable Dr. Ferti Salvan again, and all of my fellow lecturer from university in Lampung and also my beloved students from ITERA, UNPRI, and other universities. I'm very pleased to talk here today in this beautiful city, and I would like to share a bit of what's happening globally about drug discovery under practical studies, especially on the topic of osteoarthritis. I would like to talk about how the pathogenesis of this disease involved our clock, or being called by circadian rhythm, and how this pathogenesis can be potential for the strategy of the therapy. Next. Anyone in this room have a family member that experienced pain in the joint and having difficulties in move their joint during normal daily activities? Maybe your grandpa or grandma. Do you have anyone in your, do you have? Okay, yes. Yes, older people like our grandmother or our grandfather have higher risk to get osteoarthritis or pain in the joint that make them difficult to do normal daily habit. Like almost 60% of people age 80, almost 60% of people age 80 has been diagnosed with osteoarthritis. Osteoarthritis is the most common disease of the joint. Next, please. Yeah, osteoarthritis is a disease that mainly affect articular cartilage. This is the layer that covers our joint. This is the layers that covers the bones meeting in our joint. There are also other features of this disease, including osteophyte formation or bone formation in the cartilage, and also the synovial hypertrophy and also subchondral bone sclerosis. Together, these features makes the patient difficult and feel pain in move their joint. Next, please. Okay. Despite the high prevalence of this disease globally, currently there is no disease-modifying drug available. Drugs that we can find now can only relieve the symptoms of osteoarthritis. It can only relieve pain without reversing the progression of the disease. That's why the drug discovery in this disease is very famous currently, globally. Next. when we talk about the pathogenesis of osteoarthritis, and when we want to reverse the progression of this disease, it's better to look into the resident cell of the tissue affected in this disease. The resident cell, the only resident cell type in articular cartilage is chondrocyte. It has unique properties because it is called quiescent cell, meaning that it has a very low rate of matrix synthesis and also matrix degradation. However, in osteoarthritis, on the development of osteoarthritis, this cell turns into hyperactive. The chondrocytes increase the rate of matrix degradation. So the degradation and synthesis of cartilage increase. So the chondrocyte actively or hyperactively degrade the cartilage matrix and synthesis the cartilage matrix. That's what happened in the development of osteoarthritis. Later, as the disease progressed, the chondrocyte lost their ability for the anabolic process. Instead, the chondrocyte progressively degrades the cartilage. So the patient will lose the cartilage portion of their bones. So they become, they feel pain when move their joint because there is a stitch between the bones in the joints. Consistent with the pathogenesis, we can find there are several markers of this disease, meaning that there are several protein or genes that is abnormally higher or lower in osteoarthritis cartilage. They are firstly increased Indian hedgehog. This is a positive regulator for cell proliferation and hypertrophy. Secondly is a decreased level of SOX9. This is negative regulator for cell hypertrophy. And the third is increased level of cartilage degrading enzyme. So this is the enzyme that degrades cartilage layer in our joint, and the level of this enzyme in osteoarthritis condition is high, abnormally high. Next, please. Okay. So why why chondrocyte change its behavior in osteoarthritis? The potential answer is because of this circadian clock. Anyone in this room have ever heard about circadian clock? Raise your hand if you know. So circadian clock is a clock in our body. So we ourselves have clock, like what we have in this room in the wall. So we have clock. So this is the clock that we have the responsibility to synchronize the rhythm of our behavior and physiological process, and it also responsible to align our rhythm to the external cues. This is what makes we awake during the daytime and feel sleepy during the night. Next please. So the circadian clock that we have, it has hierarchical model. So it begins in our brain. So we have the central clock in our brain that is being entrained by external environment, mainly light. So light get into our eye, then it get transferred into the SGN, suprachiasmatic nucleus, in hypothalamus of our brain. So there is where our center clock is working in our brain in hypothalamus. And then the timing information here, it's being relayed to the peripheral organs through hormonals, metabolites, and neuronals. However, as research has progressed, many evidences have shown that peripheral clocks also can be entrained by external cues like feeding time or physical exercises. Next. Peripheral clocks have also been shown present in our cartilage, the layers that cover the bones in our joint that is again being affected in osteoarthritis disease. This clock is being entrained by the central clock, and it also can be entrained directly with the external cues, including sleep-wake cycle, feeding, fasting, core body temperature, and physical activity. So if we disrupt our circadian behavior, if we disrupt our circadian clock by changing our behavior daily, like our sleep time, wake time, and feeding time, we can accumulate the progress of disruption of our circadian clock. Okay. So what makes the clock ticking in our body? So the answer is there are clock genes. The clock genes is what makes the clock ticking in our body. So these clock genes are BMAL, clock, PER, and CRY. These clock genes have different level of expression throughout the day. Like PER and CRY has highest expression during the day and has lowest expression during the night. So the oscillation of the expression of these genes is what makes the clock ticking in our body. The oscillation is caused by transcriptional translational feedback loop. What is that? Transcriptional and translational feedback loop is when, sorry. I would like to share to you what is the process that makes these clock genes oscillate its level of expression during the day, that generates the clock in our body. Kursornya di daerah gambar itu, Mas, kursornya. Oh yeah, okay. So the transcriptional translational feedback loop is when BMAL and clock protein activates the transcription of PER and CRY genes. That happen during the day. That's why PER and CRY expression is highest during the day. However, during the nighttime, PER and CRY genes get back into the nucleus and stop its own transcription. That makes the level of expression of PER and CRY gene lowest during the nighttime. So PER and CRY genes expression can be entrained by the light we received through our eyes. Again, the oscillation, the different level of expression of these core clock genes during the day is what makes the clock ticking in our body. However, the length of this oscillation is actually not exactly 24 hours. Our clock is actually 24 hours and 11 minutes. So how do we match our activities with the solar day 24 hour? That's by the entrainment of the light. So the light, because of light, we can match our clock into the circulation of the earth and the sun. So the light will get into our eyes, received by retina, and then the light will be transmitted through the tract and gets into the suprachiasmatic nucleus where our central clock is present. And there, the light will activate NMDA receptor. When NMDA receptor being activated with the light, calcium will get into the cell, and calcium will activate CREB protein, and then phosphorylated CREB will activate the transcription of PER. That's how we can match our clock to the solar day, solar light day. So that circadian clock has been shown to involve in the pathogenesis of the disruption of the joint, or called osteoarthritis. How do we know that? Because firstly, the clinical features of osteoarthritis, the intensity of the pain, the intensity of the stiffness the patient of osteoarthritis feel follows circadian rhythm. The intensity is highest during the day. The intensity of the pain and stiffness is highest during the night and low during the day. So this is one of the evidence on how osteoarthritis is caused by disrupted circadian clock. Also, another interesting study have shown that people that is doing shift working, a work that requires the employment to change their working hours, like today maybe they have to work from 8 a.m. to 5 p.m. and the next week they have to change their schedule of work from 5 p.m. to 8 a.m. Like, for example, security people or nurses maybe, or hotel reception, they have higher risk to get their circadian clock being disrupted, and they have higher hazard ratio to get knee osteoarthritis compared to people that doing their job under normal daylight. So this is also the evidence that osteoarthritis pathogenesis involves disrupted circadian clock. So the evidence that osteoarthritis is caused by disrupted circadian clock can also be seen in the molecular level, at the molecular level, that the markers of osteoarthritis, Indian hedgehog, SOX9, and MMP13 activator that I showed before, they all have BMAL1 clock binding region on its promoter in their genes. Meaning that the expression of osteoarthritis marker is being controlled by clock genes per BMAL and clock. Also, the level of expression of the cartilage degrading enzyme, MMP13, its expression has a circadian rhythm, which is highest during the day and lowest during the night. This is also showing that MMP13 expression level is controlled by circadian clock. However, this can only be shown in undamaged cartilage. In osteoarthritis cartilage, the expression of MMP13 is not rhythmic. So we disrupt our circadian clock of MMP13 expression during the osteoarthritis. Also, when researchers try to knock down BMAL1 gene, they get increased level of MMP13, showing that without functional BMAL1, the core clock genes, we disrupt our joint and we get osteoarthritis. Again, another study showed that there is a difference in the expression level of PER and BMAL between osteoarthritis and non-osteoarthritis chondrocyte. So in osteoarthritis, we have higher PER expression and lower BMAL expression. So these two core clock genes being disrupted in osteoarthritis cartilage. So we come into a new strategy to find drug target in osteoarthritis that can modify the core pathogenesis of this disease. There are at least two kinds of strategy to target circadian clock in osteoarthritis that is currently being the topic of drug discovery globally. The first is by inhibiting the NMDA receptor. Anyone remember what is the role of NMDA receptor in circadian clock? Anyone? No one? Okay, it's okay. The second is the administration of melatonin. I will give very brief explanation about these two strategies. Firstly, how NMDA receptor is being targeted to find new drug in osteoarthritis. So NMDA receptor, the receptor that facilitate the entrainment of our clock to the light, is also expressed in chondrocyte cell. This shown by an experiment that give NMDA agonist into the chondrocyte cell and show that the calcium influx is higher. But when we give antagonist of the NMDA receptor, the calcium influx is lower, meaning that we have NMDA receptor in our knee that can entrain the clock there to the external cues. NMDA receptor is a receptor that has 4 subunits. Two of the first subunit is GLUN1 subunit, and the last 2 subunits can be GLUN2 or GLUN3. There are 4 types of GLUN2. GLUN2 are A, B, C, and D. Interestingly, the subunit of NMDA receptor in chondrocyte isolated from osteoarthritic patient who is doing knee surgery have different subtype. So in normal chondrocyte, we can only find GLUN2A subunit of NMDA receptor. However, strangely, in osteoarthritis chondrocyte cell taken from patient doing knee surgery, we can find GLUN2B subunit. So the subunit of this receptor is changed in osteoarthritic chondrocyte. And this change can be very detrimental because the NMDA receptor that has GLUN2B receptor, GLUN2B subunit has longer, 4-fold offset time longer, meaning that this type of receptor can get higher calcium compared to the normal one. And when researchers try to transfect GLUN2B into non-osteoarthritic cell, they get expression of PER2, BMAL, and MMP13 with the same trend as osteoarthritis. So people can make osteoarthritis model in the cell by getting GLUN2B subunit into the cell. They get higher PER2 level, lower BMAL level, and they also get higher MMP13 level by getting GLUN2B into the chondrocyte of normal cell. So this is how people realize that maybe when we design a new drug that can target specifically gluN2B NMDA receptor, gluN2B containing NMDA receptor, maybe we can modify the disrupted circadian clock in osteoarthritic cartilage of the patient. The second strategy is melatonin administration. Anyone know what is melatonin? Maybe students? Any students ever heard about melatonin? No? So this is the hormone produced by the pineal gland of our brain that has circadian rhythm. And it also plays its role to maintain the circadian clock run well in our body. So melatonin has been given to osteoarthritic mouse, and the researcher found that melatonin can modulate the expression of the core clock genes, BMEL1 getting higher and PER2 getting lower by giving melatonin. So this is the second strategy that being tried during the drug discovery of osteoarthritis by targeting circadian clock. That will be the final discussion from me. If you have any question or another input, I will be very happy. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Mrs. Apotheker Nisa. So we'll move to the next speaker. Please welcome Ms. Apotheker Dwi Aulia Ramdini M. Farm. She was born in East Lampung. Currently, she works as a lecturer at Department of Pharmacy, Faculty of Medicine, Universitas Lampung since 2019. Before, she worked as pharmacist at Kimia Pharma and lecturer at Stikes Muhammadiyah Prengsewu from 2018 until 2019, and then Universitas Tulang Bawang from 2018 until 2020. She holds her bachelor degree from Universitas Setiabudi Surakarta in 2013. Then her profession program was obtained from Unjani, Universitas Jenderal Ahmad Yani Cimahi in 2015. And her Master of Pharmacy degree was obtained from Universitas Pajajaran Bandung in 2018. Right now, she also actively involved as committee of the Indonesian Young Pharmacist Group, Lampung chapter, and also the member of the Indonesian Pharmaceutical Federation, Bandar Lampung chapter. She has published 2 books and numerous articles, both in national and international journals. Once again, please big applause to our third speaker, Ms. Apotheker Dwi Ramdini.
Thank you, Mr. Wayu, for the nice introduction. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Tabi pun. Selamat pagi menjelang siang. Ya, semoga masih tetap semangat ya. Mungkin saya akan mix ya dalam 2 bahasa, in Indonesian and in English. And okay, yeah. First of all, I would like to thank you for having me here to meet you. And the first, the Honorable the Vice Rector of UMPRI, and then the Dean of Faculty Health Science, and my fellow, and also my student here. Today, maybe you a little bit like, this is hard maybe, yeah. I would like talking about the potential drug-drug interaction, drug-herbs interaction, and their management in the clinical practice. Before we have talk a lot about development, drug development, and then the clinical trials, and also we have Miss Apotheker Nisa talking about the osteoarthritis. And now, I hope we can more learn together, and also like a discussion maybe after this presentation. So, at the first, I would like, the next, oh, sorry. Okay. This is grab a dairy product or maybe milk, a glass of milk, and then the medication. Look at when I was on a student in school, elementary or maybe junior high school, I have maybe assumption or maybe belief that we taken medication together with the milk or product dairy that will can neutral or maybe loss effect of drug. And have you ever heard? Have you ever heard about that? Yes, if yes, we have the same, right? And now, this is an introduction that we will to talking about more, because I think it's the way to give information and give knowledge to the patient. I believe mostly of here is a health professional, right? Okay, and I hope this is can improve our knowledge when we meet the patient. This is our outline overview. The first, we going talking about the definition and scope of drug-drug interaction, and then the drug interaction data in the clinical studies, and how the management DDIs or drug-drug interaction, and also the challenges and the opportunities of drug-drug interaction. Okay. Before we have explanation from the Prof. Vikas about the clinical trial, that was really a long journey of a drug, as we know. And now, it's related about the pharmacovigilance. As we know, pharmacovigilance is a post-marketing surveillance to aim to identify and quantify the risks associated with the use of drug. Then, this contributing to better understand the most important characteristic to adverse drug reaction, or maybe adverse drug event. So, from the ADR, we can see the DDI, or drug-drug interaction, is a part of the ADR, or maybe is one of factor to be ADR, right? And ADR is must be prevent in in the treatment, yeah. And then DDI are one of the commonest causes of ADR, and we reported from the clinical studies, the manifestation are common in the elderly due to polytherapy or polypharmacy, because maybe in like elderly having a lot problem, disease problem, right? So they have a much or maybe complicated to use the treatment. Okay, what is a drug interaction is? Drug interaction, yeah, this is a definition, right? Drug interaction is a situation in which drug, food, herbal, or other extrinsic and intrinsic factors affect activity of medication. We can see here, not only drug, but also food, herbal, or other extrinsic and extrinsic factor affect the activity of medication. Jadi tidak hanya obat yang akan mempengaruhi, obat dengan obat saja yang akan mempengaruhi aktivitas obat ya, tapi juga termasuk food, makanan, herbal, dan juga faktor-faktor lain. Dan ini adalah, this is a point of the drug interaction is. And then, what this implies? This effect can be increase or decrease. Increase the effect, or maybe before talking about the effect, is a plasma concentration. As we know, the medication into body and then the turn to system circulation of blood, and then after that we got the effect. And this is a explanation how the drug interaction can affect to medication with increase or decrease, or maybe produce a new effect. That's maybe the new effect is not related with to drug pharmacology's properties. And thirdly, often the efficacy or toxicity. Yeah, that's related to adverse drug reaction. We have to prevent the toxicity. Yeah, it's related to safety of treatment. Beside the efficacy, we ensure the safety when we use the medication. And then drug interaction are real. This is a real and relevant problem in clinical practice. Yeah, this is so real, and we can found directly with the patient when they have a drug interaction. And due to the increasing prevalence, while the multi-morbidity and polypharmacy. Okay, and the next one is what type of drug-drug interaction that I mentioned before. The first is drug-drug interaction, when the drug meets another drug, and then the drug, food, or beverage interaction, maybe not only the meal, maybe beverage, and then the drug, vitamin, herbal, or supplements. That's the type the drug-drug interaction can be occur. And this is a relationship among medication error. As we know, DDI or drug-drug interaction is a part of the drug-related problem. It's a part of the medication error because it's the factor become the adverse drug reaction. And generally, the drug interaction is not always related to in desire, but maybe in a little bit can be give a desirable effect, but mostly is in undesirable effect that we have to wear in this effect. And then generally the drug interaction is classified based on the underlying mechanism. The first is a pharmaceutical. It's associated or it's related to chemical and physical of a drug molecule. As we know, drugs have a characteristic of a physical and a chemical. And it can be maybe give a different drug interaction type. And then the second one is pharmacokinetic. It's related to the journey of drug in the body, including the absorption, distribution, metabolism, and the excretion. And the third is a pharmacodynamic. Pharmacodynamic is talking about how to drug molecule bind the receptor and the specific enzyme. This is classified of a drug interaction by underlying mechanism. And the first, we're talking about the pharmaceutical. Maybe it often occur in the clinical setting when we administer or preparing the administer of the drug IV, right? Yeah. The pharmaceutical is occur outside of body. It means the preparation before the administer of drug, and then generally is associated with incompatibilities of drug. It means is when we preparing or maybe the mixture of the drug before
administered to in the body. And then the manifest of this type interaction is incompatibilities, are haze or turbidity, or maybe particulates form, and then the color change. And what's the next is this effect can increasing the potential for endothelial damage. And as we know, this is not safety for the patient. And when we aware with this drug interaction, maybe the risk of the adverse drug reaction like this is not be happen or occur. And then with knowing the pH and the osmolarity of the intravenous drugs can allow their capacity to cause endothelial damage to be assessed. So we should to calculate or maybe to caution with about the pH and the osmolarity of the intravenous when we administer when we mixture the drugs. Okay, this is one of the clinical studies. This article talking about the drug interaction between ciprofloxacin and parenteral nutrition administered. And this aim is to determine the compatibility between ciprofloxacin and 18 compound of parenteral nutrition administered. And the result is, from this article, explain did not cause any color change or sign of destabilization in the fat emulsion, because parenteral nutrition in the form fat emulsion, and then precipitation, or maybe the particulates form is found in formulation higher ciprofloxacin. So this article informed to us we have to more calculate and then more aware with this preparation. Okay, and the move to the next is a pharmacokinetic drug interaction. It's a drug interaction occur when the drug alter the disposition, such as absorption and then the distribution, elimination, and also excretion or elimination. And then the pharmacokinetics interaction may result in increase or decrease of a plasma of drug concentration. Specifically in the pharmacokinetic, we will talking a lot about the plasma concentration level in the blood. Okay, the first absorption is affected about the physical chemical characteristic, and then the distribution is related to lipid solubility of a drug molecule, and then how to reach the concentration in plasma, and then the extravascular tissue, and a binding to plasma transport protein and tissue. And the metabolism is a most drug are metabolism and deliver. The metabolism, we have actor, that is a enzyme, CYP 450. That's enzyme that have a responsibility to to metabolize drug in the body. And then the excretion is a process elimination to drug or metabolite from the body. And this is the most, as we know, most of the drug in the body are eliminated in the urine. Okay. We move to example. Maybe it's a little bit example for what is drug interaction when at the absorption phase. The first is antifungal agent, such as like ketoconazole. Have you ever had ketoconazole, right? Ketoconazole and itraconazole. It requires an acidic environment for being properly dissolved. Therefore, their co-administration with the drug able to increase gastric pH. Example is PPI, such as like omeprazole maybe, or maybe lansoprazole. It can be decrease the both dissolution. Before the medication absorb, they have to dissolve that. And any antifungal agents and the other medications such as like PPI or maybe omeprazole, it can be causes interfere the absorption. And the next effect is a decrease the bioavailability of the antifungal agent. And the next one is in distribution. And the example is atorvastatin with cyclosporin. Atorvastatin, it's metabolite and it's active beta hydroxide form of simvastatin, and our substrate are the hepatic uptake transport. They have transport with ATP or organic anion transporting polypeptide, which is also inhibited by cyclosporin. That this interaction related to organic transporting polypeptide. And the fourth is drug interaction in the excretion phase. Okay. Oh. Yeah. The example is when we taken cephalosporin and with the probenecid, it can be serum level of drug affected raise. It can be increases the serum of level because related to pH in the urine or in the kidney, or maybe can be acidic or maybe the alkalinized. Okay. And this is a point important we have to know about substrate inhibitor and inducer. Maybe it's especially in the pharmacies. We have to know about it and aware about it because particular or maybe some medication have characteristic enzyme in metabolize. Substrate is are drugs or substances that which are metabolized by cytochrome, including they activate pharmacology, or maybe inactivate the metabolite of drug. And after that, to excrete or elimination from the body. And then the inhibitor are substances which compete with other drug for more than one CYP enzyme. And then when they act as an inhibitor, it will decrease clearance of substrates requiring a biotransformation for excretion. And then this may lead to enhanced effect or toxic level of a substrate. So, jadi ada substrat, ada inhibitor, ya. Substrat ini adalah obat yang dimetabolisme oleh enzim tertentu di dalam tubuh, ya. Kemudian inhibitor adalah obat yang berlaku atau dia punya peran sebagai penghambat enzim tertentu dari yang memetabolisme obat lain. And then the third is the inducer. Inducer are substances which increase CYP enzyme activity. This type is induced when the drug induces the activity of enzyme that metabolism another drug. So what's the implies? It will be will be increase activity and decrease the lethal plasma in the body. Maybe it can become the therapy, subtherapy, or maybe loss of the effect therapy. I'm sorry, it's so, so cute, so little. Yeah, this is a type of the substrate enzyme. There are CYP1A2, and then CYP to C9, and CYP to C19, and CYP2D6, and CYP3A4. And the most of the type of the enzyme CYP is a CYP3A4. Yeah. And then this is the inducer. The example, several drugs as a inducer, such as like fluoroquinolone, and then including ciprofloxacin, ofloxacin, that's the inducer, and then amiodarone and SSRI, and etc. This is an example, the drugs as an inducer and inhibitor. And then the next one is genetic polymorphism in DDI. As we know, we have talking about the metabolism enzyme CYP, and this is genetic polymorphism can be give different expression when the DDI happen. This is a hereditary of genetic difference among human being or group of people can lead to excessive. There is some, or maybe there are some race or maybe ethnicity have a genetic polymorphism, and it can be associated with the expression of the enzyme. The first is extensive metabolizer, and then the poor metabolizer, and the ultra extensive metabolizer, and enhanced enzyme expression. And it can be, you know, like influence the metabolism of drug. And then the third is a pharmacodynamic, is related to pharmacological properties effect of the drugs. When we use concomitant or together with another drugs, we have same target receptor, or maybe site, or maybe some organ system may be produce pharmacodynamic drug interaction. And it's typically categorized as a synergistic, additive, or maybe antagonistic. And this example, in this table is example additive and antagonistic pharmacodynamic interaction, such as like NSAID. When NSAID concomitantly used with SSRI, it can be possible effect increased risk of bleeding. And then the ACE inhibitor used with spironolactone can possible effect is produce the hyperkalemia. Okay, and move from the classify drug interaction in general. This is maybe the addition topic related to complementary. This is a drug herbal supplement interaction. As we know, herbal or maybe supplement is familiar, and mostly our people in Indonesia especially using a lot the herbal. And ini adalah yang harus kita perhatikan dalam penggunaan herbal. It is not always safe, not always good when we take herbal. We have to remember if the drug can be interact with another substances or maybe another drug, even the herbal medicine. Maybe the problem is the many other supplement or maybe the herb is predict to cause interaction based only on in vitro studies, they have not been confirmed. Jadi memang masih karena obat herbal itu masih kebanyakan belum melalui fase yang panjang, jadi kita agak sulit untuk mendapatkan gambaran untuk profil efek sampingnya seperti itu. And some supplement may cause interaction with a few medication, but are likely to be safe with other medications such as like curcumin and echinacea, garlic, ginseng, and the green tea. And this is a table of the herbal dietary supplement with the risk of DI or drug-drug interaction. And I will highlight with the ginkgo biloba. Maybe tidak semuanya ada di Indonesia, may not exist in Indonesia. Ginkgo biloba may reduce international normalized, potential increase bleeding risk. Jadi ginkgo biloba ini ketika digunakan bersama obat pengencer darah ini akan meningkatkan resiko perdarahan, ya. Pengencer darahnya di sini adalah contohnya warfarin. So, jadi tidak selalu, it's not always safe when we take drugs with another, even the herb supplement or maybe herbal medicine. Okay. So, how to deal with drug interaction in a clinical setting? Especially as a clinical pharmacist, we have to do some process. The first, collect, assess, plan, implement, and the follow-up. And I will highlight this is about the communicate, document, and collaborate. What is the point of this explanation? We have to collaboration with the physician, pharmacist, and the nurse to prevent the drug-drug interaction because it's real and this is not, like a simple problem. This is a part of a drug-related problem. So we have to collaborate with other health professional, physician, nurse, and maybe the nutritionist. Okay, and the next one is management of the potential DDI. In general, the first is we have to identify drug-drug interaction or maybe drug-food interaction and drug-herbal interaction. Maybe as a pharmacist, we have to aware when the patient take a medication, especially when they have a polypharmacy or maybe more than 5 drugs, we have to check. We must be check, are there drug interaction in the patient? Even just the potential we have to fix or we have to answer. And then the second is assessment risk. Assessment risk of a drug interaction is the effect is serious or maybe the moderate or significant and the minor. When we found the serious, we have to avoid. And we have to avoid use concomitantly or use co-administration. And when we found as a moderate, we have to monitoring. We have to monitoring the patient condition and maybe the clinical specific related to drug effect. And then the third is the minor. Yeah, this is a need monitoring too, even it's just minor. And also, as a pharmacist and maybe nurse or physician, we have to update data, drug interaction, especially in clinical data in patient. Yeah, this is one article that I have talking about, this is not mine, talking about the management of drug-drug interaction among critical patients with chronic kidney disease. This article showed about the pharmacists have a responsibility to make ensure the medication is safe. And the result is, clinical practice perform important intervention is timely identifying, managing DDI and prevention associated patient harm. This activity, including the identify and the managing, is give positive effect when they can prevent the adverse drug reaction or maybe including the adverse drug event, especially related to drug-drug interaction. And the last is the challenge studies about drug interaction. The first is there are not many clinical studies of the drug interaction, such as like a measurement of the level in the blood, and then in vitro studies and in animal model. And the second one is systematic review and meta-analysis data regarding clinical drug interaction is not, there's not a fewer. So maybe if we have interest to maybe investigate or maybe collaboration with do this study, it can be contribute to manage or update clinical data of drug-drug interaction. Yeah, that's my presentation. Thank you very much, and I give him back to Mr. Bayu.
Okay, thank you very much once again. Big applause to our last speaker for this first panel session. So, ladies and gentlemen, it is time for our discussion. For that, we'd like to invite again Professor Bikas and Ibu Nisa and also Ibu Dwi to come to the stage. So for the first term, I will give a chance to 3 questions. Okay, the first one, the lady one, the second one, the man with red clothes, and then, okay, Mbak yang jilbab ya. Okay. Could you please give the microphone to the participants? Don't forget to mention your name and also the affiliation, please. And for whom you will ask to.
Oke, cek. Oke, Assalamualaikum warahmatullahi wabarakatuh. Okay, my name is Mufidah. I'm lecturer in program study of midwifery in University of Muhammadiyah Pringsewu. Okay, I want to ask with Prof. Bikas about complementary method. Okay, in our vision and mission in program study of midwifery is about complementary method is Tibun Nabawi from Rasulullah SAW. I want ask about how to build interest or how to build trust in a patient in the application of complementary therapies, especially midwifery. And because most patients and even health workers is believed more in effect of curative treatment. Example for the application of Tibun Nabawi is such as honey or kurma or aromatherapy. And then I want ask one more question again. And how much effect does complementary therapy have compared to curative treatment? Okay, thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Okay, Waalaikumsalam warahmatullahi wabarakatuh. Thank you. Please, Prof. Bikas.
Yeah. So I could understand that you wanted to see that how you process for extraction for any complementary medicine. But I had said in detail that any ethnic population, when they practice for a long time, okay, and you need to generate the modern evidences. So there you have to see that, moment you ask, this has been used for a long time and how it is acting. So basically you do not know what particular dose, how long you are going to use it, what are the ingredients, and how many of them are active ingredient. So that is why you say that many times you ask the efficacy is different if you collect during winter time, if you collect during summer time, where there is a extreme weather is there. Okay, so better to collect, then you identify active ingredient. We have a lot of analytical procedure. So you can extract methanolic water, then initially you go for a, in vitro testing. We can use a bioinformatics also. So once you identify 4 active, like many of the regulator, they say that if you identify at least 4, then you can claim for a disease. Like she had presented osteoarthritis. And if you say this product is going to inhibit TGF beta, so you can say it will have effect on cartilage. So like that mechanism. So once you do that, you do in silico, then you do in vitro, and what dose it is going to act, what are the active ingredient. Later you can go for a proof of concept study with a particular model. Suppose you have a Alzheimer's. So you have different model. So you can test in that different model, like beta amyloid, okay, isoprotenolol, or streptozotocin, like that. So similarly, other disease also you can test it. Once you do that, next phase you go for a regulatory toxicity study, acute toxicity, repeated dose, long-term toxicity study. So this all depend on what clinical duration you are going to use it. So in clinical trial, when you plan for RCTs, randomized control trial, so definitely you can evaluate the efficacy as well as safety also. So that's how you establish. So normally you initially go for a pilot. Once you establish, you go for a large study, then it can be seek for an approval from the regulatory authority. That is what we have to do. Thank you.
Okay, thank you very much, Prof. Vikas. The next question, please. Yes.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Uh, Introduce myself Apri Budianto. I am lecturer at Health Faculty Universitas Muhammadiyah Pringsewu. I have any question to Prof. Vikas. Welcome to Indonesia, welcome to Lampung. I hope you very happy to Indonesia. I'm very interested about your describe about complementary therapy because in Indonesia maybe have similar with India people or society about use of herbal in Indonesia. In the points, how to provide a choice to people in Indonesia. I'm nursing. I usually give advice about use of herb, but the people usually not believe, about preparation of nature better than of practical of preparation. How way to increase the belief in those society on people to use of herb. Thank you very much, Mr.
Thank you for the
Vikas.
question. I think what is we practice is anything you wanted to establish, you need a data. Okay? So if you say this particular herb or particular product is working, so you need to generate the data. And it should also incorporate in standard treatment guideline. Now here we have a two choice. One is modern medicine, another is alternative or complementary system. Like in India, we have a modern and we have a Ayush. So it is up to the patient, they can opt for a alternative therapy or they can go for a modern medicine. Like suppose I am a doctor for modern medicines, I am not allowed to write the prescription for alternative one. He or she has to go to Ayush doctors. So that need to be done. But one thing is very important that when you should not take a concomitant medication. Many people, they do not inform the doctors. They have already used the alternative therapy. So as she had presented, there could be drug-herb interaction or it could be clinically significant drug-herb. So that need to be, once you inform, then you see, is there any possibility of interaction or dose dumping? So accordingly, as I said that everything comes based on evidence. So if you can generate the evidence, this is a well-established therapy, and at the same time, we have to also see that is there any personalized variation? we talk about now individual variation. One particular drug is effective for myself, not for other person. So that also you have to look at, like ethnic population, is there any genomic variation is there for efficacy and safety? That also you will have to establish. Thank you.
Okay, thank you very much, Prof, because for the explanation.
Jadi bicara data, Pak Apri.
So the last,
Bicara
yes.
data. Datanya dulu, baru kita
Microphone, please. The microphone, please, to our participant. That one, Pak. Yes.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Good morning, ladies and gentlemen. So I have one question, but before that, I would like to introduce myself first. My name is Ismano Febri. I'm student college of University of Muhammadiyah Pringsewu and Faculty of Health. So I have one question for Professor Bikas Medi about the complementary that previously you explained to us. I'm curious, sir, is it important for nurse to learn complementary therapies? And in your opinion, what are the benefits that nurse can get by learning complementary therapies? So that's my question. Thank you, sir.
Okay, please. What is the important for students to learn the complementary therapy? Yeah.
Thank you for the question. I think there are a lot of scope is there. I'll tell you. Because we do not ever, like if you say today food, medicinal food, it's a huge industry. I did not show the slide. Nutritional supplement, total market in the world is $33 billion. Okay, so earlier people used to just get an approval, even a food product or herbal product also. Now there's a particular system that you have to generate all the steps, start from in silico, in vitro, proof of concept, then regulatory toxicity study, then clinical trial. All phases, phase 1, phase 2, phase 3, and then only get an approval. So what does it mean? That all this also need to be manufacturing in GMP condition. All the industry has to create a GMP condition. So that means it is a big scope for a student to go into nutritional division, to go into complementary medicine. There is a huge industry is there. So course scope is very high. So we can be trained in that particular system, and you can have an alternative one also, like a diploma course. So that can be done, and you can easily get absorbed in industry in academic institute. Thank you.
Okay, thank you very much, Prof. Bikas. I think we still have a time. Could we give more opportunity to the participants? Okay, we'll open the second term to, okay, for 3 questions, please. Ibu and, okay, Mbak, nanti wait a minute ya, after the first one. And one more, mungkin mahasiswa laki-lakinya, ada yang mau coba tanya? Silahkan. Boleh bahasa Indonesia, nanti kita coba translate ke bahasa Inggris. Oke? No one? Oke, please Bu. Oh Ibu, oke nanti.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. I will introduce myself. My name is Dewi from Sumatra Institute of Technology. I would ask question for Professor Bikas, because I have same interest in medical drug medicine, herbal medicine. Okay. My question is does India have any specific focus in drug development using herbal medicine? Because as we know in Indonesia, we focus on finding herbal medicine related to diabetes and hypertension. So my question is, Does India have any specific focus on drug development using medicine, using herbal? Like that, thank you.
But what I could hear,
Can you
can you
get
repeat
okay?
Is India have any specific focus in drug development using herbal medicines? Does India have any specific focus in drug development research, focus research related to disease?
The research of Prof. Vikas?
No, in India as general. In India as general. Gak kedengeran ya? Di India, secara general ada nggak spesifik apa namanya penelitiannya untuk beberapa penyakit misalnya?
Okay. The question is there any specific research that focus on the some diseases? Yeah, in India.
as you said that you are interested in pharmacology, thank you very much. And as you know that India is a large country. So we have national importance for a particular disease, like for malaria, like for filaria, tuberculosis, leprosy, all the disease, we have a national program. Now, if you talk about tuberculosis, we have introduced all the new drug, like bedaquiline. It has been implemented. So we have, program for communicable disease, and we have a program for non-communicable disease. Because over a period of last 2 decades, diabetes, hypertension, all these diseases also coming up, like celiac disease, okay? Other places it is not there. So there is a task force for that. So all the funding agency, we have a Ministry of Health. Under this, we have ICMR, Indian Council of Medical Research. Then Ministry of Science and Technology, we have DST, Department of Science and Technology. We have DBT. We have a big organization called CSIR, Central Scientific Industrial Research. So there is a huge number of organizations. They also collaborate with all across the globe. And we have all the program for communicable as well as non-communicable disease, national program. Thank you.
Okay, thank you. Actually, I have one more question because related to the homeopathy. Because we know that there are 2 strategies for medicine, therapy of medicine. The first is allopathy and the second is homeopathy. So I saw on your presentation before that Ayurvedic related to homeopathy. Do you have any specific reason why in India focus on homeopathy? Thank you.
I think what I said is, we now have a complementary integrated department. It is called Ayush. A for Ayurveda, okay? And then U for Unani, okay? Y for Yoga. Like that, we have an integrated course, Ayush and homeopathy. And recently, it has been introduced a program called AMSI. So all, they have a different division. Ayush, basically, they look at all the traditional product. They evaluate it scientifically, okay? Then yoga, the yoga has been spread across the globe. So we have an integrated called Ayush. Now they have created a ministry called Ayush. So all the medical school, like recent medical schools, they have a different branch called Ayush. So people can go to modern medicine, people can go to Ayush. That's how it has been divided. Yeah.
Okay, I think enough. Thank you very much. For the next question, please. Okay, Mbaknya, silahkan. Microphone, please. The microphone, please. Over there. Please.
Assalamualaikum warahmatullahi wabarakatuh. The Honorable, the lecturer, all the guests and all the speakers, thanks for the MC who give me opportunity so I can ask this question. Okay. I want to ask a question towards Prof. Vikas. Because Prof. Vikas was talk about Ayurveda treatment. So my question is our DEVA same with yoga treatment because Indonesian people more familiar with yoga treatment and which benefit more between all of them. Thanks for the
Yeah, I think there are a lot of herbs that is used, but it is not with yoga is, with the healthy people or other, they can use. But when somebody falls sick, then only you are going to use some product, medicine. Otherwise, not altogether. There are a lot of nutritional product is there, but you have to also see that how long you can use. Because any product it is used, you have to see that how long it is safe. It is not all the time you are going to take it. So normally we advise that if we use for 6 months or more than 6 months, we need to have a long-term toxicity studies for the rodent 2-year studies. And if it is there, then you can say, you can go ahead. But any product you use, there should be a monitoring mechanism, safety monitoring. So that should be there. It should be with under supervision. Okay, it is nothing like OTC, over the counter. It should be there. Thank you.
Okay, thank you very much, Prof. Vikas. So the last one, Ibu, please. Microphone.
Okay, thank you. I want to ask to Mrs. Dwi. Okay. Let me introduce that I come from Economic Faculties of UMPRI. I have one ask that how long is the human body can withhold to chemical drug?
How long?
Berapa lama tubuh
Yeah,
manusia bisa bertahan dalam meminum obat-obatan kimia? Okay.
So your question is how long human body tolerate the chemical?
Yes.
Okay. Like I say, I wanted to use a particular product for 10 days. Okay. How to justify? I am going to use for 10 days. I'll do a studies in rodent. I'll give it a single application, observe the animal for 14 days. Then you sacrifice, we take out all the vital organ. So it will give you the indication from acute toxicity study, what is the level of NOEL. NOEL is no adverse effect at that level. And you also evaluated LOEL. LOEL is low adverse effect that level. So from that, you get an idea that this dose is adequate to get an effect. Okay. Then second point, it also give an indication that if it is overdose, how you are going to manage in the emergency. Similarly, if you wanted to use for 25 to 30 days, we have a data generated for 90 days, 180 days, 6 months, 1 year. This is called repeated dose toxicity study. So whatever NOEL and LOEL you are getting, you are confirmed in repeated dose toxicity study. That means if you use this drug for 10% of the lifespan, how the body is going to behave. Similarly, when you talk about hypertension, diabetes, any chronic neurological disease, you take the medicine for years together. So there you go for a long-term toxicity study. But when you use for more than 6 months, you are going to ask the question, can it cause mutagenicity? Can it cause carcinogenicity? Because when you chronically use drug, it is remain there in the body for a long time. So you need to ask that, can it cause cancer or can it cause mutagenicity? That is what you do a battery of tests, mutagenicity and carcinogenicity. Then only you approve, you can use for 6 months, 1 year, 2 years like that. So in a clinical trial, you verify that. Thank you.
Okay, thank you very much. And maybe Bu Dwi would like to add something?
Okay, maybe menambahkan ya Ibu. Mohon maaf Ibu dari ekonomi dengan Ibu siapa Ibu? Ibu Iin. Ibu Iin. Oh yeah. Yeah, thank you for the question. Maybe the explanation from the Prof. Bikas about how to chemical drug in the body. Maybe saya mau menambahkan ya, jadi untuk berapa lama obat itu di dalam tubuh, begitu Ibu ya? Di tubuh kita.
Bertahan.
Ya, Pak? Oh yes, it's related to tolerate, ya. Jadi berapa lama tubuh kita akan bertahan dalam menggunakan obat seperti itu, Ibu? Ya, iya. Tentunya yang pertama adalah setiap penggunaan obat itu berbeda tujuan, ya. Jadi tergantung pada penyakitnya. Kemudian ini juga akan berpengaruh kepada kondisi tubuh dalam mengkompensasi dari penggunaan obat, karena sudah kita tahu tadi bahwa obat itu akan banyak berinteraksi dengan bagian dalam tubuh kita, termasuk hati, and then the kidney, ginjal, seperti Ibu ya. Jadi kalau pertanyaannya apakah tubuh kita akan kuat, ya selama kita akan memang harus membutuhkan obat itu, maka dia akan mampu, karena tubuh kita memang sudah didesain untuk bisa menerima chemical walaupun itu memang obat. Tapi karena memang kondisi kita membutuhkan obat tersebut, ya ini berkaitan dengan risk and benefit Ibu ya. Jadi mungkin arahnya kepada bahwa obat orang minum obat takut ginjalnya rusak seperti itu Ibu, mungkin ya. Kaitannya seperti itu. Iya, mungkin itu tergantung kepada tujuan dari penggunaan obat itu sendiri gitu. Tapi sebetulnya tubuh kita memang mempunyai kekuatan untuk bisa menerima obat itu sendiri selama dosis tadi ya Prof. Bikas sudah menyampaikan bahwa dosis yang digunakan itu sudah atau sesuai dalam range terapi seperti itu. Mungkin Ibu Nisa mau menambahkan?
Mbak Nisa mau nambahkan cukup? Oke, thank you very much. I think time is up, so please big applause for all our speakers once again. Yeah, thank you very much. And we believe that there are many questions remained, but still we don't have time anymore, so we have to close this first panel session. And before we move to the next panel session, I would like to thank you once again for your participation, and I do apologize for any mistakes, and don't forget to join our second panel session after lunch break. And at the end, I would like to thank you very much, and wassalamualaikum warahmatullahi wabarakatuh. Thank you very much.
Okay. Thank you very much for all the speakers and also the moderator for giving such an informative and comprehensive discussion. Please kindly stay on the stage because we would like to present a ceremonial gift as a token appreciation for sharing your valuable knowledge in this session. And to present the ceremonial gift, we would like to invite the Vice Rector of Academic, Dr. Triyuni Hendrawati, MPD, the Vice Rector for Human Resource and Finance, Nurse Asri Rahmawati, MKes, the Dean of the Faculty of Health, Mrs. Elmi Nuryati, MEPit, PhD. The appreciation gift and certificate will be given by Dr. Triyuni Hendrawati, MPD, to Professor Bikash Mehdi. Iya. Nurse Asri Rahmawati will give the appreciation gift and certificate to Dwi Aulia Ramdini M. Farm. And then Mrs. Elmi Nuryati M.Epit PhD will give the appreciation gift and certificate to Nisa Yulianti Suprahman MSc. And also Mrs. Elmi Nuryati M.Epit PhD will give the appreciation to Mr. Bayu Anggileo Pramesona PhD. Okay. Okay. Stay on the stage because we would like to take pictures together. For the Vice Rector of Academic, Vice Rector of Human Resource and Finance, the Dean of the Faculty of Health, all the speakers and the moderator, thank you. Okay. Ladies and gentlemen, this end of the first session. And we will go for afternoon refreshment. We will have short break, and after that we will back for international symposium at 1 p.m. Thank you. For the co-host, please sit on your chair because we will give you for lunch. Thank you.
Cek. Baik, Bapak Ibu peserta simposium bisa mengambil makan di depan ya, silahkan. Nanti kembali lagi jam 1, on time ya, akan diabsen lagi. Bapak Ibu co-host, monggo silahkan juga bisa beristirahat terlebih dahulu, nanti bisa kembali lagi jam 1. Terima kasih.